Why me? What did I do wrong? Will I ever be okay? I feel so alone. In our digestive system, food travels a long journey. It gets broken down and absorbed by our bodies to nourish it. For most of us, this is a simple process. But for people with inflammatory bowel disease, it is a long journey filled with obstacles.
I tried a bunch of different drugs. It went from local administration to like steroids to immune suppressants, and then eventually I was hospitalized due to sort of like a major flare up, where I then had to have surgery and have my whole colon removed.
IBD refers to a group of disorders that cause chronic inflammation in the digestive tract. There are two main types: Crohn's disease and ulcerative colitis. This inflammation isn't just painful, it's disruptive.
In May 2nd, 2008 was my first emergency surgery where, the doctors, it was basically life or death. If we don't do surgery now, my intestines would rupture and, I could become sepsis and go into a coma and all of that.
When people hear IBD, they often think that it's just spending more time in the bathroom, and that's far from the whole story. This disease affects every part of life.
There's so many other ways that it can affect your body, and just from your oral health to your joints to your eyes to your skin. There's just so many other ways it can manifest, and a lot of people don't
And
realize.
while some medications can help manage IBD, they come with significant side effects. For some, these side effects can feel just as debilitating as the disease itself.
Developed compression fractures in my spinal cord due to long-term use of prednisone in my top vertebrae and my bottom vertebrae. I had liver issues. I was followed by infectious diseases as well because your immune system becomes so compromised.
The concept of our project was to design a cost-effective, patient-friendly bacterial treatment for IBD with a focus on shifting from traditional methods like injections and pills to a more comfortable inhalation therapy. Previous studies have shown that lactic acid can inhibit overactive immune cells, helping to reduce inflammation in autoimmune diseases. We wanted to apply this for IBD patients in a way that minimizes discomfort. To do this, we transferred the muscone receptor from mouse olfactory cells into yeast and created a muscone molecular switch. This switch activates the yeast to produce and secrete lactic acid when triggered by muscone, which can be inhaled by the patient. We also knock out the original receptor in yeast to stabilize the system and ensure that it functions safely. A two-component system allows the bacteria to specifically target IBD-related substances in the intestines, ensuring safe colonization and removal when necessary. Finally, we developed a comprehensive model to predict the effectiveness of this treatment, giving us precise control and optimization of the system. Let's explore how we brought this concept to life.
Our wet lab is divided into two parts: the therapy system and the colonization system. First, let's talk about our therapy system. We wanted to create a muscone-induced lactate secretion system. We divided it into two parts: the lactate secretion and the muscone switch. After ensuring that each part worked independently, we combined them. The first part focuses on lactate secretion. We introduced the exogenous lactate dehydrogenase LDH gene into yeast cells to facilitate lactate production. The gene is driven by the GAL1 promoter, which is induced by galactose. We tested the modified yeast by providing either galactose or glucose to measure how much lactate was produced. The yeast with the new gene produced more lactate when induced with galactose, confirming the system was functioning as expected. However, we observed unexpected lactate secretion even under glucose induction, which can be seen in the graph on the right. This led us to suspect background expression of the LDHA gene. To address this, we redesigned the experiment, adjusting for differences in yeast concentration and controlling induction time more strictly. The results confirmed that carbon sources significantly influence lactate production. After these adjustments, we confirmed that galactose indeed led to higher lactate secretion than glucose. Let's talk about the second component of our therapy system, the muscone molecular switch. We engineered yeast to express a muscone receptor along with a signaling protein controlled by a galactose-inducible promoter. When muscone is present, it activates a downstream signal that can be detected through fluorescence, acting as a switch to indicate system activation. When we induced muscone and observed the cell under a microscope, we found that muscone significantly increased GFP fluorescence, confirming the switch was working. Then, combine the lactate secretion system with the muscone molecular switch. Now, the yeast can produce lactate when the muscone receptor is activated, creating a complete therapeutic system. We tested this integrated system by exposing the yeast to muscone under galactose induction and measured lactate production. The results confirmed that muscone successfully triggered lactate secretion. The second part of our wet lab is the colonization system, which helps engineered yeast specifically target inflamed areas in the small intestine. This system has two key parts: a sensor that detects chemical signals in the intestine, and a protein that helps the yeast stick to the intestinal lining. First, let's talk about our sensor, TTRSR. It detects tetrathionate, a chemical signal that increases in areas of intestinal inflammation, therefore making it the perfect marker. When the yeast senses tetrathionate, the TTRSR system activates and triggers the expression of downstream genes. To test the effectiveness of the sensor, we designed plasmids that express a fluorescent protein EGFP in response to tetrathionate. We transformed yeast with these plasmids, then induced some with the tetrathionate and compared the fluorescence intensity between the induced and control groups. The yeast successfully produced the fluorescent marker when tetrathionate was present, confirming that the sensor worked. However, the fluorescence was weaker than expected. We believe this is because the sensor proteins may not be functioning optimally in yeast due to differences in how bacteria and yeast process proteins. To improve this sensor, we plan to optimize the TTRSR protein, focusing on codon optimization and ensuring the sensor proteins are properly located in the yeast membrane. This will strengthen the yeast response to tetrathionate and make the system more reliable. Moving on, let's talk about the second component of our system, ALS3. For the yeast to stay in place in the inflamed areas, we use the ALS3 protein, which is found in the Candida albicans. ALS3 helps the yeast to stick to the intestinal cells by binding to E-cadherin, a protein found on the surface of these cells. This binding ensures the yeast remains at the inflammation site long enough to deliver treatment. We designed an experiment using human intestinal tissue sections to test whether yeast expressing ALS3 adhered better than yeast without ALS3. We stained the tissue sections with blue dye to highlight the cells and introduced yeast with and without ALS3. Yeast expressing ALS3 shows significantly better adhesion to the intestinal tissue compared to the yeast without ALS3. However, some issues with inconsistent fluorescence made counting the yeast cells difficult. To improve the consistency of the results, we plan to use a more reliable cell dye such as trypan blue for better staining and tracking. We're also exploring alternative adhesion proteins to further enhance the yeast's ability to stick to the inflamed tissue.
The main goal of the hardware is to make it easy and effective to deliver muscone. We've come up with two ways to do this.
We use a nebulizer to release muscone. The nebulizer turns a muscone solution into vapor, which can fill in air or be inhaled directly. A small microcontroller called STM32 is used to control it, and it can run for a long time while plugged into a power source. There is a button on the device that lets you control how long the vapor is released, helping to manage the amount of muscone inhaled.
The second part of our dry lab is our mathematical models that help us understand the pathway of our therapy. It's divided into 4 interconnected parts: muscone inhalation and distribution in the body, muscone receptor binding at the cellular level, intracellular signaling cascade activated by muscone, and lactate absorption model. We created a compartment model to stimulate how muscone, once inhaled, travels through the body and reaches the intestine. Firstly, it enters the alveoli or the respiratory mucosa, permeating to the pulmonary microvessels into the bloodstream and is distributed throughout the body. It reaches the intestine through the mesenteric network, where its concentration increases. This simulation shows that after one breath, traces of muscone begin to appear in the intestine, confirming the therapeutic concentrations can be reached. We then perform molecular dynamic simulations to visualize how muscone binds to its receptor, and we analyze the receptor-ligand interactions. These simulations confirmed that muscone binds effectively to the receptor with stable interactions observed at key amino acid sites. Once muscone binds to the receptor, it activates a cascade reaction in the engineered yeast cells. Using ordinary differential equations, we simulated this process, focusing on the phosphorylations of protein like SCE5, SCE11, SCE7, and FUS3. This ultimately led to lactate production. The cascade reaction was simulated, confirming that yeast cells efficiently respond to muscone, activating the pathway necessary for lactate. Our final model addresses the core question: why is our lactate-secreting yeast system superior to direct lactate administration? We developed a compartment model to compare the sustained release of lactate by yeast to direct ingestion. The model showed that direct administration results in a rapid decline in lactate concentration, while induced secretion provides a controlled and sustained lactate levels over time, preventing adaptation and excessive lactate buildup.
In human practice, we bridge the gap between the lab and the real world, focusing on 3 key areas: engineering, economy, and social impact. First, we address the core issue of our project, engineering success. We consulted with experts, including biology professors, nanomedicine specialists, technicians, and IBD professionals. Together, we refined the concept of treating IBD through inhalation. Next, in the economic aspect, we conducted comprehensive research on the pros and cons of existing IBD medications. Our visit to Lanjing, a manufacturer in Shanghai, inspired us to turn our project into a marketable product. Regarding the social impact, we considered the relationship between our product and the society. We conducted 7 patient interviews across diverse demographics and integrated their feedback into our work. We also consulted IBD doctors from Southeast Asia and China about the precautions necessary for our treatment to align with official medications.
In our project, biosafety is a top priority. First, we engineered yeast that require specific nutrients to survive. These yeasts have deficiency in 4 essential nutrients: lysine, leucine, uracil, and histidine. Without these nutrients, the yeast cannot grow outside the lab. We've also developed a suicide system that activates when the yeast leaves the intestines. Outside the gut, there are no bile acids, so the yeast produces a protein called MAZF, which causes cell death. Inside the intestine, bile acids trigger a protein called FXR, which in turn activates Cl protein. Cl blocks MAZF, allowing the yeast to survive. Since bile acids are only found in the gut, this ensures that the yeast can't survive outside, ensuring safety.
At iGEM Tsinghua, we prioritize inclusivity by making synthetic biology accessible to everyone. To do this, we created educational content in multiple languages. We hosted workshops and produced videos that explain synthetic biology in simple terms. Additionally, we made our wiki accessible for people with all kinds of disabilities. And we provided a booklet explaining our project in multiple languages such as French, Arabic, English, and Chinese, including an audiobook version by QR code for those who have difficulty reading.
As we move forward with MuScure, we know that there's still much to improve. Our next steps focus on refining our therapy system and enhancing our models. We're grateful for all the support we've had and for the patients who've given meaning to our work.
Living with pain and uncertainty of IBD shouldn't be the norm. IBD patients should live fully and enjoy time with their loved ones. While there is still a long road ahead, we hope to contribute to a future where suffering is no longer the norm.
MuScure, inhale, heal, repeat.
Live a full, happy life with IBD, and that it is part of you, but it's not all of you.
That you're not alone and somebody else is going through this, and that, it's okay.